I think it's better than vlogging. So I think and I don't want people to know.
What's your name?
Greg.
Nice to meet you.
Nice to meet you.
Where are you from? Where are you out of?
I'm a consultant. I work out of West Virginia.
Oh, okay.
I was in the Loafman Lab.
Oh, okay. All right. It's crayfish legacy then.
I've wanted an auction. I've never been to a derby, and I apparently
How you doing? I didn't see you there. When did you get in? Oh, okay. Cool. Well, you missed the first half, but I recorded it. I'm under a grant obligation to make some videos and stuff, so that's the main reason I'm talking here.
Hey Dave.
Hey, Stretch.
Thanks for doing this again.
No problem.
For those who were here on Monday, which most of you guys were, David Hayes has graciously again offered to give another presentation on Boone's Crawdads.
All right.
So it should be good. Thanks a lot.
Thanks, Stretch. Thanks for enduring another one. Like I said, I got a small little grant award from EKU, and part of this is I have to make some recordings and outreach videos and stuff, and so I don't like talking to just my computer or in my basement, so it's nice to be able to have at least a captive audience that knows something about this stuff. So last time I talked about sort of the setup here and these crayfish that are sort of inhabit areas that were part of historic trails, such as the Wilderness Road that Daniel Boone had traveled. So just real briefly to review, since some of you weren't here, I'm convinced that the area that you're in is the most interesting area of Kentucky because it is the meetings of the headwaters of several major rivers. So we have the Cumberland River, the Green River, the Rolling Fork, the Salt, the Dix, and tributaries to the Kentucky here. So this is sort of a meeting place. And this is likely driven by historic faulting in this area. And so this is a map with the faults overlaid with historic trails such as the Wilderness Road, the Warrior's Path, and you are right here. And so you've got this intersection of these pathways and trails. And the general idea is that maybe the Wilderness Road was a paleo drainage, essentially that humans simply found the path of least resistance. And I won't get into it, but part of my reasoning for that is because of some of the genetic diversity we find on the other side of Cumberland Gap. And so compared to things over here. So the idea is that maybe the crayfish have been here before the uplift or the most recent uplifts, not the original uplifts, but the more recent uplifts. And so to have all this data from a class that I've been teaching, Principles of Molecular Biology. I get them to generate barcode data on crayfish. The ones around here are ones that I've collected over the years, but I've also included ones from other previous Mudbug manias and other places in the state. So we have several hundred barcodes, and won't get into the complexities of this like I did last time, but I'm happy to answer any questions. So feel free to ask me to come back to this slide later on. But one of the things that we found in that project is juvenilis seems to have introgressed with everything. So I mentioned last time, it seems like Faxonius juvenilis originated in Buck Creek. The oldest haplotypes for juvenilis are in Buck Creek, which is right about in this neighborhood here. And so it seems like when it got out of Buck Creek, it had sex with everything it could find. And its mitochondrial DNA continued upstream after it made contact with these things, such that you get populations of Christovarius in far eastern Kentucky, say stretches collected from Hell from Certain Creek, and you will get a mix of both Cristovarius haplotypes and Juvenilis haplotypes, even though only Cristovarius is there. Morphologically, they all look like Cristovarius. And so it seems like it doesn't seem like it's this constant hybridization. It seems like this is an occasional thing. And then once the mitochondria gets into that other lineage, it's continuing to move upstream. And so it's moved, it's introgressed. This is the main introgressor. It's mated with every other one of these Faxonias. And so it got me thinking again, not just about Boone, but then maybe crayfish evolution in general. And maybe not just the Faxonias, the rusty crayfish and Kentucky River crayfish that I've been talking about, but some broader questions. So I started reviewing old crayfish literature, really old crayfish literature. I really got hyped in on Huxley's study of zoology, the crayfish. He marketed this as a zoology textbook, and then when you open it up, it's only about crayfish. It was like, sort of a scam in a way. Darwin had prompted him to write a popular zoology textbook, and Huxley said, okay, I can teach everything somebody needs to know about zoology by teaching them about crayfish. And so the book is like, says study of zoology, and then you open it up and it's like, oh, this is all crayfish. But I got really into it. I found a copy that Dr. Schuster had left around, and I picked it up, and I feel like there's a lot of gems in there. I really highly encourage you to find a copy. It's also available digitally. And it's kind of like, Darwin, reading it is kind of a slog, but there are these gems sort of buried in there that you got to kind of find. And Darwin's gems have all been found, but Huxley's gems have not been found, except by me. I've found them now, and I have them flagged and stuff. And eventually I'll share some with you. But it's also a good brain exercise to make yourself sort of sit down and understand that Victorian language. So I highly encourage you to check it out. I've also, Huxley talked about North American crayfish in here, particularly species of crayfish from Kentucky. And his main reference, though, was this Herman Hagen from 1870, the first monograph of North American crayfish. I was talking to Dr. Schuster, and he told me that Hagen was actually a German immigrant who was hired by Agassiz. And Agassiz at the museum, the Harvard Museum, basically gave him a jar of crayfish and said, here's your project. This is your work. And this was the only crayfish work he ever did for the rest of his life. So this was like a contract work for Agassiz. And so this is about 100 years later after Boone emerged. I don't talk about it, but 100 years after this, Günther Schuster described Cambarus batchi. Weird, but all right. So I've got a couple of Daniel Boone quotes in here. So curiosity is natural to the soul of man, and interesting objects have a powerful influence on our affections. Indeed, Daniel Boone. And so this is the plate from Hagen of the gonopod morphology. The unusual thing about North American crayfish is this morphological diversity. Nowhere else in the world has this amount of morphological diversity in their crayfish reproduction. You'll get one or two, and they're a little bit smaller, and some of them don't even have them. But this is one of the real mysteries of North American crayfish, is why does North America have this complex diversity in reproductive morphology? It's also unique to North American crayfish in that they call the curious dimorphism of North American crayfish, where they're going through these Form 1 molts and Form 2 molts. Now my audience here knows all about this, that the Form 1 is sort of the reproductively active molt, and the Form 2 is the sort of reproductively inactive molt. I sometimes will say this is their cycle between Chad and incel. So Form 1 is their Chad, Form 2 is their incels. They flip-flop back and forth. And in the springtime, they're in Form 1, but they're not mating. And then they go into the summer into Form 2, and they're not really doing much of anything. And in the fall, they get back into Form 1, and that's when mating occurs. And so it seems like this flip-flop molting is related to seasonality, possibly hydrologic pulses. But we don't really know why. The other weird thing about North American crayfish is the I don't have it on here yet, but this is the annulus ventralis of the females. This is also a unique structure to North American crayfish. This is almost like a little pocket that holds a packet of sperm, which we will then use later to fertilize her eggs. that pocket is also unique to North American crayfish. And it's sort of a blind pocket. It doesn't go internally, but they are incredibly complex. There are a variety of angles to it. Some of them are very narrow, some are very open. Some are left, some are right in the same species sometimes. So the annulus is also very complex. But for now, I'm just talking about the gonopods here. Quick adjustment. And so here's a more modern representation of gonopods, photo by Dr. Schuster there. Here's my annulus ventralis photo. This is all one species, and if you looked close at this, you'd be able to see that some of them are left and some are right. We don't really know why. Some of them are left and some of them are right. So again, I guess that's 3 mysteries total: unique morphological diversity in males, unique diversity in females, and then the cyclical form 1 and form 2. The third other sort of anomaly is these ischial hooks. So the hooks at the base of the legs that we think sort of help them lock together for mating, but we're not quite sure too. This is unusual that these features have only appeared in North American crayfish. So that got me thinking about crayfish sex. And so I realized that we don't really know what's going on in there. When these two are clasping together, we don't really know what's going on because these organs are not ducted organs. So when we don't call them a penis because a penis is a ducted organ. Plus, these were named in Victorian times, and that was a dirty word back then. So they had to think of a different name for all these structures for every single one. But it's not ducted. Sperm is transferred externally down a groove, and it comes out somewhere down here of that leg, not at the end. And so these are not inserted into the female. There's something else going on here, and we don't really know because it's hard to see what's going on there. And so, again, we don't, I sort of blew the question here. We don't really know exactly how they work. Again, it's used to transfer sperm down a groove. The sperm comes out an opening of the leg, and somehow it is transferred down this groove and meets somehow got transferred to the annulus ventralis. And these projections here, these processes, some of them are short and some of them are long, and we don't really know why. But we do know it's not a lock and key. All right, so they don't necessarily fit together like we think sexual parts usually fit together, and the lock and key and the biological species concept. But we know it's not that. Had ideas about maybe these were for scraping sperm from competitors, but that doesn't really explain some of the environmental influence that we see. If they are just for sort of scraping out the packet of another sperm, from the sperm of another individual, why would we have this huge morphological diversity in them? Classic paper in this field asking or combining genetic data with looking at gonopod morphology, Taylor and Knopf 2006. Won't go through all this, but basically this was the first attempt at what we now know as Faxonius and a few other genera to look at the evolution of this particular characteristic, the male, form 1 male gonopods. And the main take home from this, that I get from this paper, is that sympatric species that live together, tend to have similar gonopods. Sister species, that each other's closest relatives, generally don't co-occur with one another. They're generally allopatric, and they have different gonopods. So things that are close related look really different, but then things that are in the same area start to look similar. Why? We don't really know. This is the first crayfish paper I was asked to review by Dr. Schuster years ago. I probably will butcher it, Trisolescens. They were getting ready to publish this, and Dr. Schuster's like, will you take a look at this data? I don't know what's going on here. I was brand new faculty at EKU. I think this was around 2013. And I was like, I don't know anything about crayfish, but then I look at the paper and I was like, send me the data and send me like your Excel tracking files and stuff. So I wasn't able to provide any sort of groundbreaking information on this paper, but I was able to plot their sites out, and I noticed they have this category here of short and long gonopods for this whole big complex. And so note the narrow differences between what they're calling short and long. I mean, that's short and that's long. So these are very small differences between these two. But this is a whole species complex, and this isn't the main character for identification in this group. But they basically said, all right, we'll just call which ones are short and which ones are long. So I plotted that characteristic out in the map instead of their species, and I apologize for the color here, but this is what I saw, where my green dots here are long gonopods and red here are short. And here you see the fall line, essentially when you get off of the plateau and you get into the Gulf coastal plain. You can see it's not perfect along it, but it's pretty close. It seems like these long gonopods are up on the plateau, whereas these shorter ones are down off of it. Why? I don't know. There's this other paper that I think is really interesting and provides a model for a lot of the stuff I've talked about in the previous Boone's Crawdads on Faxonius in this area. I think this is a model of exactly what's going on in Kentucky, but it's a much more simple system because it doesn't have all of those faults and complex paleo drainages that we see in Kentucky. And so these species aren't necessarily sister to each other on the tree. But they are nested within this broader complex that I'm talking about with Christoverius, Juvenilis, Rusticus, those things. So these are in there. These are the Eastern Ozark relatives of this group. And so if you notice here in the distributions, we essentially have two species here in the Spring River and one in the 11 point. And in the 11 point, you've got, which one is this? Wagneri. Here we've got Robert I in the Spring River, and Eupunctus is sort of up further on the Ozark Plateau. And you'll notice some of these gonopod differences between these two, or between these three. Now again, they're not each of those closest relatives on the mitochondrial tree, but they are somewhat closely related. So it got me thinking about this idea that maybe the gonopods reflect some sort of historical uplift. Rather than modern day uplift, perhaps they're reflecting uplift. And so I mentioned you have to think about the cross section of Kentucky because these rocks were sort of laid on top of one another. So the bluegrass at one time was hundreds of feet higher than it is today, right? And so we have to think about the landscape a lot differently than what we think about the landscape today. So I went back, I was thinking about, all right, what do these gonopods do? Why does it seem to be related to habitat or where they're found or stream gradient or the height of historic mountains? Why is it related to that? And so I'm trying to make sense of the diversity. And I went back to this paper and I said, and I'm thinking about Boone and exploration, and I thought about, well, what if I thought about like the exploration of crayfish, and started thinking, what are the first crayfish that were found? The first crayfish that was described was Camberus bartoni, type locality is probably around Philadelphia somewhere. And then I was like, well, what if I just looked at the crayfish that were found in Kentucky? So that's what I did with this Hagen paper. I pulled out all the species that were in Kentucky. Again, Boone's Crawdads, and these were the first crawdads that were known from Kentucky. Now I did I cut two out a little bit later for good reason, but let's just say we pulled out and we just looked at these individual lineages from Kentucky. And then I started lining them up. And so what we have here, Programmerus clarki, Orconectes pellucidus, Camberus bartoni, Faxonius immunis, Faxonius placidus, Faxonius rusticus, and Faxonius juvenilis. These were the first Kentucky crawdads, essentially. Now, I'm not saying all the type localities were in Kentucky. I'm saying these are the ones from that paper that we know occur in Kentucky. Now, I knocked out Procamberus acutus because it's pretty similar to clarki there, and then the pictures just got too cramped. So I cut out a few just for aesthetics purposes here. And so I started thinking about these, all right? Does this say something about humans traveling across the landscape? I mentioned the first one described, Camberus bartoni. Pellucidus is mentioned in Huxley's book because when people first found about cave crayfish, blind crayfish, this was really interesting information to folks. So he specifically mentions those. And so it started thinking to me, well, where were people going to find these things? And so I don't have these in taxonomic order. I do need to do that. What I want to do is say this was first described and then this. But so essentially think about Procamberus clarki, the red swamp crayfish down in Louisiana or far western Kentucky it gets up to. It's mostly in swamps, ponds, wetlands, things like that. We've got organisms in caves. Canberra's Bartonii. I'm sort of using this for like all Canberras and Canberras-like things, but they're found in burrows, also springs and rivers. Immunus tends to be in slower, often smaller rivers, but sometimes larger, but generally slower systems. Placidus tends to be in big rivers. Rusticus, medium big rivers. Sometimes it'll get into lakes, but lakes aren't really part of its natural, it's more of its invasive habitat. And then we have Faxonius juvenilis, which you've all become familiar with. It's found from the tiniest little tribs to the Ohio River, right? And so you sort of get this sampling of habitats across the state, which I thought was interesting. And then I said, I could make a tree, I could make a phylogeny of this. And so I've made giant phylogenies of all Canberridae with Co1, and I was able to just pull those out and say, all right, let's look at the sequence of these. Which I was like, well, all right, it doesn't have all of the crayfish in, all right? Not all of the crayfish species that we know. It gets much more complex when we add them in, but can we come to any sort of conclusions by just this limited data set? And you see Procamberus clarki as most basal here. Then we have Pellucidus, the cave species, Bartonii, Immunus, and then essentially our Faxonius here. And so is there sort of a general trend here that we can get from this reduced complexity? It's almost like Taylor and Knopf was too complex for us to sort of get some of these patterns out. Because it is really complicated. This is what the whole data set looks like in an unrooted tree. It's huge, it's a mess. And so, again, I'm thinking about this across the landscape, and I start plotting out where these things are. So Procamberus clarki out over close to the Mississippi embayment. We've got Pellucidus, the cave species, down in Mammoth Cave area. We've got Immunus out in western Kentucky. Now Bartonii, for Canberras, I should really have this spread across the entire state because this lineage, or this gonopod type, which is a representative for us, is everywhere essentially. But for this purposes, we'll leave it over there. And then we have our longer Faxonius. This is Placidus. This is Rusticus, and then we have Juvenilis here. So I'm like, what's going on here from this cross section? So there's this gradient, or maybe historical gradient, but still, why gradient? So I'm in my garden one day. This is a picture of me from Google Street View. When I first moved here, we didn't have Google Street View. I could not view Google Street View to look at the house I was buying, and so the next invasion is the surveillance state. There is no escape from the surveillance state. But anyway, I'm working in my garden, clawing at the landscape like crayfish do, thinking about the juvenilis in my backyard here, the ortmanni, the occasional tenebrosus on my neighbor's property down here. And I'm pulling weeds there. And I'm thinking about these things. I'm thinking about the landscape. I'm thinking about all this crayfish gonopod morphology. I'm thinking about Daniel Boone and all these faults and stuff like that. And a car drives by, and it's, some shit box. And I'm like, startled, and I look up, and suddenly a whole bunch of Mythbusters episodes came back to my mind, and I thought, maybe they're spoilers. And so this car, and so I remember these Mythbuster episodes of like illustrations about what spoilers do and drag and things like that. Got me thinking about NASCAR here. And so I've got a comparison here. Two guys here. We got Richard Petty, known as the King, here on the left. Over here we got Dale Earnhardt. Richard Petty, the King. Dale Earnhardt is the Intimidator. And one difference, this is from about the 1970s. This is the early 2000s. Notice the main difference here is this spoiler. So for a very brief period of time, Richard Petty and other drivers had these really high spoilers on their cars. And it was great for driving, but they are almost getting too much lift. And so NASCAR changed the rules. So you got to knock that down. The cars are going too fast because NASCAR is an endurance contest. 500 miles, but they don't want people to die because if they're going to leave and they're not drinking beer or things like that. So they want to keep the race going for that 500. And so they're constantly changing the rules and changing the landscape, and the drivers have to adapt to it. So they said, no, you can't have that high spoiler. And so over time, they switched to this really stubby spoiler. And this changed the physics of driving, and that you have Dale Earnhardt emerge as the Intimidator. His driving style was much different from Richard Petty. The two overlap. These are also both the highest winning, they have the highest number of wins. Two overlapped for briefly, but they obviously weren't racing in different cars here. But Dale Earnhardt, his driving style was to ride up behind somebody, almost bumping them, or actually bumping their bumper, hence the name, the Intimidator. So the guy would be like driving and Dale Earnhardt's in your rearview mirror, and he would use the draft, essentially, the reduction in air pressure and turbulence behind the previous vehicles, or the vehicle in front spoiler, and use it to slingshot around. Essentially use the physics to make him go faster around the car. And the only way he was able to do that was this modification of the spoiler. Drafting was much less efficient when you had this wing type spoiler. So thus, the landscape of NASCAR was changed by the rules, and the vehicles adapted in response to it. And so, got this idea that maybe gonopods are spoilers, all right? And that their main job is possibly to deflect flow around the female's annulus ventralis. And so this could possibly explain why the length of these things are influenced by stream gradient or habitat or things like that. Essentially, not quite elevation, but slope possibly. Now we don't know for sure, but again, this is transferred externally. It's coming down a groove. If you're looking at the animal from the top, essentially the annulus would be down there somewhere. Here's a representation of the annulus. So the idea is that maybe this thing is sticking in front of the annulus, and maybe these crayfish are sort of facing in the flow. Remember, it's during the fall, during increased hydrologic pulses. And so perhaps they're facing in the flow, and thus the gonopod length may change in relation to velocity. And so I've got like a rough model here. So you have this faulting event, right? So you have an uplifted area, you have a high area and a low area, and eventually it's going to erode down. But this steeper area may select for longer gonopods, but not just gonopods, but also longer snail shells, or sometimes more rounded snail shells like Bleptoxus. And so I've got these ideas of how this works out for Pleurocerods too. But over time, without consistent uplift, this is eventually going to erode down, potentially flat, like we see it today. Like I said, I drive over faults all the time on my commute. They're all flat now. So eventually this is going to decrease. And over time, this is where you're going to get your increased community complexity. And so it could be that this faulting is, modifying velocity and flow. And then, and essentially we're seeing that in the bodies of the invertebrates, and particularly the fluidics of crayfish sex. So we have this diversity here. It might be driven by these faults. And so here's an illustration that I meant to have on a previous slide there. But you can see a short spoiler versus a tall spoiler creates much more, the red here is low turbulence. And so these other colors are really high turbulence. So you see it creates this pocket of low turbulence. And the idea is that these gonopods are doing the same thing and have changed as the uplift has eroded down. So it might be that the ancestors of, say, Cambarus, with these sharp curved ones, may have actually been longer and pointier. They've been here for a long time, but as the landscape eroded down, they might have become curved in response to erosional forces. So I'm thinking about like, all right, how do we actually quantify this? I'm working on getting some 3D scans of it, of a gonopod. So far, nobody's done that yet. If anybody wants to beat me to it, I'm having a heck of a time. But I think once we get some 3D scans of gonopods, we can do a whole lot of cool things. But I started thinking about like, all right, how am I gonna do this data analysis? What's the math gonna look like here to actually prove or at least support this hypothesis? And it takes me all the way back to Isaac Newton, and essentially Newton's second law of motion, which is force equals mass times acceleration. Now, this has been expanded out to take into account velocity, viscosity, things like that, fluid dynamics. And all of fluid dynamics boil down to something called the Navier-Stokes, or the Navier-Stokes equations. And so this is a group of equations that we use to solve a whole lot of things. To solve how fire will move through a building. We use it to explain or to design airplanes. We use it to sort of figure out weather patterns, to predict weather. This is why it's hard to predict weather, essentially, is because this math is really difficult to do without sort of fudging it. We kind of The Navier-Stokes is one of the $1 million math prizes, essentially, for a couple reasons. But one of the reasons is that we kind of have to make assumptions to figure it out. It's really hard to figure out the true sort of path, because if you think about what Navier-Stokes is trying to do, it's trying to say you drop a drop of water in a river here, you're going to be able to calculate essentially where it will end up and exactly its path. And we know that's very difficult to do, if not impossible. So we make assumptions like laminar flow. When you think about cubic feet per second or something, a lot of those, and discharge, you're assuming laminar flow. Those are the assumptions that we do to be able to do those calculations. But laminar flow doesn't actually happen in nature, but we don't do the calculations because it's much more complicated to do the actual calculations to figure out where that might go. And so it could be that this Navier-Stokes equation, again, is you're using it to predict the rivers and courses of water, but also the formations of galaxies. So essentially anything that you have that you want to describe turbulence in a system, you're going to use the Navier-Stokes equations. And then I started reading about the Navier-Stokes equations and the physicists and what they have to say about it. So here's a quote from Richard Feynman: "Turbulence is the most important unsolved problem of classical physics." This one from Werner Heisenberg: "When I meet God, I'm going to ask him two questions. Why relativity and why turbulence? I really believe that I'll have an answer for the first." When you start hearing the physicists talk about God in your data analysis, you know you're onto something good. And I've literally heard multiple physicists talk about how to know the Navier-Stokes equation is to peer into the mind of God. Because if we could actually do it, well, it would mean you could predict the future, right? And this is why it's impossible to actually get to the true Navier-Stokes. The idea that you could drop a particle of water and to be able to predict the future of where it would end up is very difficult, right now impossible. Now people do this kind of math, and you have to do tons of simulations. I'm talking to a guy who's in our fire science program, and he does little fires in a box, and he looks at the smoke, and he does 1 million iterations of his model. It's computationally intensive. My father-in-law does, he's an HVAC engineer for like big commercial buildings. And he's like, oh yeah, we just had to have somebody do computational flow analysis for us, and it's gonna cost us $50,000 to do it for ductwork. Now I'm talking about doing this on a microfluidics level, and then so even if I say it's, yeah, all right, these are spoilers, but then linking it to past uplift events is going to be a difficult job, and I haven't figured out exactly how we're going to do it yet. And so again, I just brought this up again here to show that perhaps these are little spoilers deflecting current and are being shaped over time by the uplift and erosional processes across the landscape. I've also this is something called the energy cascade of the Navier-Stokes. So with force times mass acceleration, that force. What's the force? We're talking about uplift. So that force is going to introduce energy into a system. So this chart is showing the sort of response after that energy input. So you have energy in, and it creates large eddies. You get big eddies at first, and then you have the energy cascade, the inertial range, it sort of slows down, the energy's out, and you get little eddies. Interestingly, Thomas Huxley talks about the whirlpool of life, and he loves this analogy of the whirlpool of life. And this is essentially what this is, because you're getting eddies here, big eddies, and they get gradually smaller, 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 and smaller, sort of spiraling down this energy curve. And so it could be that the faulting across the landscape has caused this such that we might be able to plot species on this energy curve. So Faxonius putnami has the longest gonopods, and it might be at the top of that energy curve with that Mississippian plateau. And then we see these other lineages that are sort of further down that landscape. But then we have the mitochondrial DNA that seems to mostly move upstream. So it's like the powerhouse of the cell is continuing to push upstream while erosional forces are pushing downstream and shaping the sex lives of these crayfish. Now this is the landscape of Kentucky flipped. I have it mirrored here so that it kind of looks more like this energy cascade. Again, I don't know exactly how we're going to tie these things together, but this is sort of the big idea here, so that the gonopods and the annulus evolved as sort of microfluidic traps, right? So that they were able to survive the highest periods of time. So remember, Lexington was much higher. the Kentucky River crayfish originated in the Cumberland River in Buck Creek and likely went up essentially the Lexington Dome that has since eroded away into the flatter bluegrass region that we know today. So again, the gonopods don't reflect current day habitats or geology. They reflect historical uplift and historic velocity, and it's almost like they're vestigial. Like they don't necessarily have to be able to withstand the fastest moving water ever. Kind of like humans. We are the running primate. We are adapted for running. Our head is on a pogo stick. I don't run for anything. So just because they have them, and maybe this was the selective pressure for them, doesn't mean that they actually need them now, and it doesn't necessarily mean that they need this velocity for actually mating. Like the sperm transfer may be good enough in order for them to get some sperm over. But the idea is that these more extreme historic environments are what actually shaped the reproductive morphology. So I started thinking, I was like, well, I have this big tree of Cambarus, and if this is, or Cambaridae, and if this is what's going on, where's the most basal member of the family? So I'm sure Brooke will maybe correct me when they get bigger data sets, bigger genomic data sets, but this is the one I think it might be, Faxinella blairii, Blair's fencing crayfish. So if you look at the gonopod of this thing, I mean, it's nuts. It has two cross gonopods like fencing swords, hence the name Blair's fencing crayfish. I've noted a lot of weird coincidences. When I got to this one's especially weird because my wife's name is Blair. And so I was like, maybe she's like, they're telling me to pay attention to my wife or something like that. The other weird thing is that when I was a grad student, I worked down in the system in the Little River, and I worked on mussels there, and it's one of the most biodiverse rivers in the United States. It's like almost every species of mussel you could name as a different species because there's a genetically unique haplotype in the Little River in this area. And so why would this one be it? I've lost my figure here. What this would show is that this lineage occurs right on the edge of the Ouachita thrust line. So the Ouachita thrust line used to be continuous with the Appalachian Mountains until the Mississippi embayment was created and sort of divided it up. And so this lineage occurs in a place that has likely been uplifted and eroded, uplifted, eroded, uplifted, eroded multiple times throughout its history. And the end result is this completely jacked up gonopod with a really long end that reflects the steep periods of uplift and the really short end that reflects the sort of the lower gradient time. So it's adapted to an environment that is changing, even though we associate it with an environment that is very flat. Sorry, got my things out of order here. So I'll just put them all up. So just to review, the idea again, admire the faults. So this process of faulting creates this energy cascade, which again, we can sort of explain with turbulence and Isaac Newton and Navier-Stokes. And the idea here is that this space here is the place where information transfer is happening. That's where sperm is being transferred to the female. And it's this process, these three, across this landscape has led to this diversity because of reproductive success at this point in the fall under the leaves. So I got one last thing here, one slide here. Again, I'm on a Huxley kick. So I got a quote out of here. This is from the 3rd page of his book. Again, I highly encourage you to check it out, but I'll read this here. In relation to the human mind, nature seems boundless, and though nowhere inaccessible, she is everywhere unfathomable. Thank you very much.